Welcome. So today we're diving into the incredible story of T-cell mediated immunity. And this isn't just about any old immune response. Nope. This is about the body's elite special forces, the specialist cells that are trained to hunt down and take out invaders that are hiding where nothing else can get to them. So how does this all work? Well, let's get into it.
I mean, it's a really fascinating problem when you think about it, right? Antibodies are fantastic for fighting enemies out in the open, in your blood or in your tissues. But what happens when a virus, or even some bacteria, actually breaks into one of your own cells and decides to set up shop? How will you fight an enemy that's essentially wearing your body's own uniform? Well, that is the core mission of our T cells.
So we're basically going to follow the entire life story of a T cell. We'll start with its fundamental mission. We'll see how it gets the green light to act. We'll peek inside to watch that chain reaction of commands, and then we'll witness the huge army it builds. And finally, we'll understand how it actually wins the war and remembers the enemy for life. All right, let's kick things off with Section 1, the mission. To really get a handle on our T cells, we first have to understand the unique kind of enemy they are specifically designed to fight, the intracellular microbe. Okay, so this is the central challenge. You've got viruses, certain bacteria, like the one that causes tuberculosis, and even some fungi that have all figured out this really clever strategy. They don't just attack, they invade. They hide out inside our own cells. And this makes them completely invisible to antibodies, which can only patrol the areas outside of cells. So to fight them, the immune system needs special agents that can recognize an infected cell from the outside and eliminate it. And that agent is the T cell. So how does a T cell even get the go-ahead to attack? This brings us to a really critical moment, activation. And listen, if you're studying for an exam, this is one of the most important concepts to really master. This is the moment a totally naive, inexperienced cell gets its orders. Here we go. These are our two main characters in this story. On the left, we've got our naive T cell, totally ready for action but pretty inexperienced. And on the right, you had the professional informant, the antigen-presenting cell, or APC. Now, the T cell uses its T cell receptor, that's the TCR, to inspect the antigen the APC is showing it. Its co-receptors, CD4 or CD8, they help make that connection nice and stable. And then there's another molecule, CD28, just sitting there, waiting for a second, absolutely crucial signal. And that second signal has a name. It's called co-stimulation. Honestly, the best way to think about it is as a safety check. Your immune system does not want its T cells, its elite killers, launching a massive attack just because they see some antigen. It needs confirmation that this antigen is part of a genuine danger, like an actual microbe. This is what prevents the immune system from making a terrible mistake and attacking our own healthy tissues or other harmless things. And this right here brilliantly illustrates the two-signal hypothesis, which is a fundamental principle of immunology. You see, signal 1 is the T cell receptor recognizing the antigen. But if that's the only thing that happens, the T cell either does nothing, or even worse, it enters a state of permanent unresponsiveness called tolerance. To get a full-blown activation, for it to multiply and specialize, it must receive signal 2. And that comes when the B7 molecule on the APC binds to the CD28 receptor on the T cell. So, signal 1 plus signal 2 equals go for launch. To really lock this concept in your head, just use this analogy. Activating a T cell is just like launching a missile. You need two separate keys, and they have to be turned at the exact same time. The first key is the antigen, that's target identification. The second key is that co-stimulatory signal from the APC, that's the confirmation of hostile intent. You don't have both, there's no launch. It's a simple but incredibly powerful safety system. Okay, so both keys have been turned. The T cell has received its activation orders. But what happens next? What's going on inside the T cell? Let's take a look at the biochemical chain reaction that's just been set off. Whoa, okay. At first glance, this looks incredibly complicated, kind of like the circuit board for a supercomputer. And, it is. But don't worry, we are not going to get lost in all those details. The important thing here is to understand the logic of the process, which we can actually break down into a pretty clear, manageable sequence. So here's the crucial point. When the receptors on the outside of the cell bind to their targets, it switches on a series of enzymes inside the cell. These act like a line of falling dominoes, a chain reaction. This signal travels all the way to the cell's command center, the nucleus, and it activates these special proteins called transcription factors.
Just think of these guys as generals who can go in, unlock the cell's DNA, and give the order to start making a whole new set of genes, the battle genes, that are going to transform this naive cell into a full-on warrior. Activating those battle genes unleashes two massive changes. First, the T cell is ordered to build an army through huge proliferation. And second, all those new soldiers get trained to become specialists, each with a very specific role to play in the fight ahead. And just to appreciate the scale of what's about to happen, consider this number. Before an infection, the number of naive T cells that can recognize one specific piece of one specific microbe, it might be as low as 1 in a million, just a single cell. But through a process called clonal expansion, that one single cell, once it's activated, divides and divides and divides again until it has produced an army of over 100,000 identical clones, all of them armed and ready to fight that one specific enemy. It's just an incredible mobilization of forces. This graph here shows the whole campaign. See? The infection starts, and within days, the number of T cells that are specific for that microbe just explodes. That's our clonal expansion. They do their job, they eliminate the threat, and then, once the war is won, the numbers drop off sharply in a phase we call contraction. And this is really important for returning the body to a state of rest. But notice, the number doesn't go all the way back to zero. A small group of veterans is left behind. And that army isn't just some mindless mob either. The cells differentiate into specialized units. The CD4-positive T cells become helper cells. Some act like battlefield commanders, secreting chemical orders to direct the whole attack. Others become what we call follicular helpers, and they stay in the lymph node to help B cells make better antibodies. And meanwhile, the CD8-positive T cells, they differentiate into cytotoxic T lymphocytes, or CTLs. These are the direct assassins that hunt down and kill infected cells one by one. So the battle is won. But just as important as knowing when to attack is knowing when to stop. The final phase of the response is all about control, hitting the brakes to prevent damage, and memory, making sure the body is prepared for any future attacks from the same enemy. Okay, this is another one of those high-yield comparisons for any exam. The immune system has two main brakes, CTLA-4 and PD-1. CTLA-4 acts early, back in the lymph nodes, and it basically outcompetes the CD28 go signal to keep that initial activation in check. PD-1, on the other hand, acts later, out in the peripheral tissues where the battle is actually happening. Its job is to calm things down and prevent too much damage from chronic inflammation. So, two different brakes for two different stages of the response. And this brings us back to those veteran soldiers that are left behind after the battle. These are the memory cells. They can survive for years, sometimes even decades, kept alive by these little maintenance signals. They don't really do much during peacetime, but if they ever encounter that same enemy again, their response is exponentially faster and way stronger than a naive cell's ever could be. This right here, this is the entire basis for long-term immunity and exactly how vaccines work. So let's just end with this thought. We've seen how the body uses these brakes, like CTLA-4 and PD-1, to shut down T-cell responses and prevent autoimmunity. But what if a T-cell's target isn't a microbe, but a cancer cell? See, cancer often learns how to hit these brakes itself to avoid being destroyed. So what if we could block those brakes and just release the T-cells to do their job? This very idea, it's called checkpoint blockade, is the foundation of Nobel Prize-winning cancer immunotherapy. It's one of the biggest medical breakthroughs of our time, and it all comes back to understanding these fundamental rules of how a T-cell gets activated.